Now, the next thing to know is that we're going to talk about glucose catabolism. We're going to talk about breaking down glucose quite a lot, but that is not the only method. That's not the only kind of organic molecule that can be used to get energy. We have all of these different kinds of macromolecules. You know that proteins are polymers of the amino acids. So if we use a lot of hydrolysis reactions, we can break up proteins into the individual amino acids, and then we can start working on them, getting energy out of them as well. This will happen if you're really overdoing it in terms of dieting and exercise. There's no sugar, no fats, no glycogen for your body to turn to for energy resources. It'll start catabolizing your proteins, your muscles instead. So watch out for that. But this is also a good process when you are eating protein. If you eat a whole bunch of, say, almonds, you get some protein going into your body. Well, then your body needs to be able to break down those proteins into amino acids in order to be able to build up your muscles if you're working out. Carbohydrates obviously can be broken down into sugars, glucose, for example. We're going to talk about that quite a lot. But fats also can be broken down. We use fats to get energy. You're going to remember fats, one of the purposes of them is long-term energy storage. We're going to break them down into their fatty acids and into glycerol. Now, what this is showing right here is that you can actually break down these other kinds of molecules into some of the constituents that we're going to see later on in this chain. So for example, amino acids, if you deaminate them, if you remove nitrogen from them, you can actually convert them over to pyruvate, which we already mentioned is the product of glycolysis. And they can enter the mitochondria, they can enter this metabolic pathway at that point then. You also have fats. They can be broken apart, and you can get some of the other intermediate products from those fats. Remember that fatty acids are mostly just carbon-hydrogen chains, hydrocarbons, just carbon to carbon to carbon to carbon to carbon with a whole bunch of hydrogen on the outside. So it's pretty easy to get something sugar-like by breaking down a fatty acid. In fact, the constituent that is shown here, pGAL, glyceraldehyde 3-phosphate, G3P, you might remember that from the end of photosynthesis, I hope. It's the product of the Calvin cycle. So you're starting to see some of these same familiar molecules starting to show up in cellular respiration that you would have, that you saw in photosynthesis as well. Okay. Both proteins and fats can be converted into a molecule called acetyl-CoA, and that's another step in this process. And once they're converted in, they can all enter the citric acid cycle, which is another name for the Krebs cycle. What is the point of this image? It's easy to get locked into the idea that cellular respiration is all about sugars, breaking down sugars. And then you start to think, well, wait, hang on, don't we eat other kinds of food and get energy out of it? And yes, we absolutely do. But we're going to talk about the pathway for sugar. And then this image is showing that at several points in that pathway, these other kinds of molecules can be fed in. Some other metabolic process can occur which chemically changes, say, proteins or fats sufficiently to allow them to feed into this process. All right, so here we have the head honcho of cellular respiration, the big important organelle that we're going to be talking about for the rest of this presentation, the mitochondrion. The job of the mitochondrion is to do all of the parts of cellular respiration that are aerobic, all of the parts that require oxygen in order to proceed forward. So actually the first step of cellular respiration we already talked about, but here it is in a animation, right? We get glucose. There's glucose right there. It's got 6 carbon atoms. Those are the dark circles that chain down the center. It's got 6 oxygen atoms. Those are the red ones. There's 1 on each glucose there. And then it's got 12 hydrogen atoms. So C6H12O6. Glucose in the cell, inside the cytosol, not in the mitochondria yet, is going to undergo glycolysis to break sugar, glycolysis. So when it does that, we snap it in half and we get 2 pyruvate molecules. out of that process. In addition to the pyruvate, we also get 2 net ATP. As I mentioned before, we have to actually invest some ATP in glycolysis in order to get any kind of energy out. You gotta spend money to make money, right? So here we spend 2 ATP and we get 4 ATP back, so the diagram shows you having 2 net ATP. Then, those 2 pyruvate molecules, they are allowed to enter the mitochondria. So everything that happens after this point in cellular respiration is inside the mitochondria. But all cells are able to do glycolysis. And it makes sense that this would happen outside of the mitochondrion, because remember, bacteria don't have mitochondria, right? Archaea don't have mitochondria. They still need a way to get energy, though. So this anaerobic process, this is what's happening inside those organisms that are unable to deal with oxygen. Not all bacteria are obligate anaerobes, but some of them are. All organisms are able to do this, and since not all organisms have mitochondria, it kind of makes sense that this process could happen out in the cytosol, out in the cytoplasm of the cell. So those pyruvate molecules, they're able to cross over the outer membrane of the mitochondria, and in comes these 6 oxygen molecules coming in, and out comes 38 ATP, well 36 additional ATP, as well as carbon dioxide and water. So this is the overall chemical equation of cellular respiration, right? When we're saying C6H12O6 plus 6O2 gives you 6 CO2 and 6 H2O, this is what we are actually referring to. Just this molecule with all of these carbons and hydrogens and oxygens enters, we break it apart and the 2 parts enter the mitochondria. We bring in some oxygen, we rearrange all those atoms, and these products fire out. But we also release a lot of energy in that process. So, step 1 of cellular respiration is this glycolysis process. The overall reaction here is that you start out with glucose and you end up with 2 pyruvate molecules. All right. Now, in that process, you actually get 2 net ATP, as I said, but you also get this other coenzyme. All right, so we're going to keep a running total here. Glycolysis gave us 2 ATP net and 2 other coenzymes. This is another dinucleotide coenzyme called NADH. Keep track of these because we're going to use them at the end of this process. At the very end of cellular respiration, you're going to say, boy, we've made a lot of NADH, but we have not actually made all that much ATP. We're going to get the energy out of the NADH at the very, very last moment. It's a big payoff. You'll love it. But that's where it's going to be. So right now, these are 2 energy-carrying coenzymes that came from the breakdown of glucose. Okay. Now this actually occurs in 2 different phases. Well, there's a lot of steps along the way, but we can categorize them into 2 phases. The first thing you actually have to do is invest your 2 ATP, and then you have this payoff phase. So you have the energy investment phase and the energy payoff phase. Energy investment, invest 2 ATP. Energy payoff phase, you get 4 ATP back, which is why it's net. The actual process of glycolysis occurs in quite a few steps. Each step here, going from glucose at the beginning to pyruvate down here at the end, requires an enzyme. There's an enzyme here, it's called a kinase. There's an enzyme here, it's called an isomerase. There's an enzyme here, and here, and here, and here, and here. An enzyme at every single step. This is a metabolic pathway, where the products of every chemical reaction become the substrate for an enzyme to catalyze a new chemical reaction in this process. Now the good news is you do not need to memorize this entire list of reactions. We're going to simplify it pretty darn substantially. But I just wanted to show you around here. So 1, we have a step here where we go from glucose to glucose 6-phosphate. This is accomplished by using up an ATP molecule. ATP gets converted to ADP, adenosine triphosphate, 3 phosphate groups, to diphosphate, 2 phosphate groups. So this just lost a phosphate group, and that phosphate group ended up on our glucose. Breaking down glucose is a little bit like trying to solve a Rubik's Cube at times. Sometimes you have to switch groups to the back, move them around, get everything into the right orientation so that the enzymes actually can get purchase on them, can actually solve that riddle, that can actually break apart that thing. It requires a little bit of a chemical do-si-do in order to make it work out. So the first thing that we do in cellular respiration, which is the process by which we break down sugars in order to make ATP, the first thing we do in that process is use up some ATP. This is called the energy investment phase. The energy investment phase. You're investing, right? You take some money, you put it into an index fund, you wait for it to mature for a couple of years, you get a lot of money back. So after the energy investment phase, later we're going to have what's called the energy payoff phase, where we get more ATP back. But first, you got to spend money to make money. ATP is the energy currency of the cell. That's our cellular money here. So we've got to spend ATP to make ATP here. All right. Now, we break it down. We change it again into another molecule. We break it into two molecules. From this point forward, we have two of everything. And at this point, we use another ATP, 1,3-biphospho. There's two phosphate groups in this molecule. So again, we take an ATP, it loses a phosphate, donates it to these molecules here, and we get an ADP. Okay. Now, we're going to start peeling these phosphate groups back off, and we're actually going to get more of them back than we began with. So 1,3-phosphoglycerate to 3. So here we start what's called the energy payoff phase. Second part of glycolysis is the energy payoff. We have two phosphate groups here. We're going to go back down to 1. Where'd that phosphate group go? We took an ADP and we made an ATP. But now there's two of them, so we actually ended up making 2 ATP in that process. Down here, we go from phosphenylpyruvate over to just regular old pyruvate. So notice that we've lost the prefix, the phospho prefix here. So we're getting another ADP converted to ATP. And again, since there's 2 of them, we end up with 2 right there. Now, I told you in photosynthesis, when we made ATP at the end of the light-dependent reactions, that's called photophosphorylation, right? Because we used the energy of sunlight in order to make ATP, in order to phosphorylate an ADP molecule here. And we're doing it again. We're phosphorylating this. We're adding a phosphate group to this coenzyme to make ATP. But this is not photophosphorylation. This is another kind of production of ATP called substrate level phosphorylation. Just like when we talked about photophosphorylation, I'll mention the 3 different types of phosphorylation we'll talk about this year. Every time you're making ATP, we call it phosphorylation of some sort. Photophosphorylation is in the light-dependent reactions. Substrate level phosphorylation happens in glycolysis. It happens again in the Krebs cycle. And then we have oxidative phosphorylation, which will be the very last step of cellular respiration.